All right, welcome. Today we are diving deep into a disease that, when you talk about sheer devastating impact, is really second only to malaria. It's a chronic, debilitating condition caused by a parasitic worm that has one of the most complex and, frankly, fascinating life cycles on the entire planet.
So let's just start with a number. 250 million. That is the estimated number of people right now infected with schistosomiasis. It's one of the most widespread of what we call the neglected tropical diseases. So yeah, this isn't some rare, obscure illness. It's a massive global health crisis that's basically hiding in plain sight.
And all of it, is caused by this tiny parasite, a macroparasite to be precise. It has evolved this unbelievably intricate strategy for survival. It's a journey that takes it from freshwater into a snail and finally into the human bloodstream.
Okay, so let's meet the culprit. Its scientific name is schistosoma, but you'll probably hear it called the blood fluke. And as we are about to see, this is absolutely not your average flatworm.
You know, to really get what we're dealing with, we need to know where it fits in the animal kingdom. Schistosoma belongs to the phylum Platyhelminthes. You might know them as flatworms. Now, this is a huge group, and it includes everything from harmless little things in ponds to other infamous parasites like tapeworms and, of course, other flukes.
But here's the thing. Even among its fellow flukes, schistosoma is a total oddball. On the left, you see a typical liver fluke, something like Fasciola hepatica. It's broad, kind of leaf-shaped, and it's a hermaphrodite. It has both male and female reproductive organs. Now look at the blood fluke on the right. They are slender, almost thread-like, which is a perfect adaptation for living inside our blood vessels. And here's the most incredible part. They are dioecious, meaning they have separate sexes. The larger male actually has this groove where the more slender female lives, and once they find each other, they are mated for life. It's a strategy that just supercharges the reproductive output. It's wild.
And this whole lifelong partnership, it's all in service of one single goal: reproduction. And that brings us to their incredible journey, a multi-stage life cycle that is completely dependent on two things: freshwater and a very specific accomplice.
Okay, let's walk through this absolutely bonkers transformation. It all starts when an egg, which is released from human waste, finds its way into fresh water. Inside that egg is a life stage called a miracidium. It hatches, and using these tiny little cilia, it swims around looking for a very specific type of snail. Once it finds one and gets inside, it transforms into something called a sporocyst, which is basically a parasite factory. The sporocyst multiplies asexually, just cloning itself over and over, producing thousands of the next infectious stage, the cercariae. These are tiny fork-tailed swimmers that burst out of the snail, turning that water into a literal minefield for any human that happens to be nearby.
So what happens when one of those thousands of cercariae actually finds a human host? Well, this is where the story shifts dramatically, from the water to deep inside the human body.
This is crazy. The cercaria just burrow directly through unbroken skin. As they go in, they shed their little forked tails and transform again, this time into a stage we call the schistosomula. These young worms get into the bloodstream and basically take a tour of the circulatory system. They eventually wind up in the liver, and that's where they mature into adults, find their lifelong partner, and get ready for the final part of their mission.
Now this is super important. We're not just talking about one kind of schistosoma. Let's look at the three main species that infect people. Schistosoma monsoni and Schistosoma japonicum like to live in the mesenteric veins that surround the intestines. But Schistosoma hematobium prefers the vesical plexus, which is the network of veins around the bladder. You can actually tell them apart just by their eggs. S. monsoni has a big lateral spine, S. japonicum has a tiny little one, and S. hematobium has a really distinctive terminal spine. But just look at the number of eggs per day. I mean, monsoni and hematobium are pretty busy, but S. japonicum is on another level entirely. We're talking up to 3,500 eggs every single day. And notice, each one needs its own specific snail host to complete the cycle: Oncomelania, Biomphalaria, or Bulinus.
And this right here, this brings us to what is maybe the single most important point of this entire explainer. See, the adult worms are sneaky. They can live for years, mostly hidden from your immune system. The disease we call schistosomiasis isn't actually caused by the worms themselves. No, it's caused by the body's absolutely violent reaction to the millions and millions of eggs they produce.
Now, the disease shows up in two main ways. First, there's an acute phase, often called Katayama fever. You tend to see this more in people who are new to an area, like tourists.
It can start with cercarial dermatitis, you might have heard of it as swimmer's itch, from the parasites first getting in. That's followed by fever and a big spike in a type of immune cell called an eosinophil, a condition known as eosinophilia. But for people who live in these areas, the disease is a slow, chronic burn that develops over years, as their immune system is just locked in this relentless, never-ending battle with a constant barrage of eggs.
So how exactly does the body fight back? Well, its main weapon is something called the granuloma. About half the eggs manage to get excreted to continue the life cycle, but the other half gets stuck in the body's tissues. Since the immune system can't get rid of them, it does the next best thing. It tries to wall them off, forming these little masses of immune cells around them. That's a granuloma. And while it's a protective response, just imagine millions of these tiny granulomas forming over years and years. That is what leads to the tissue damage, the scarring, or fibrosis, and all the really severe symptoms of the disease.
And the damage is very specific to where the worms are living. For S. mansoni and japonicum, those granulomas in the gut wall cause pain and can even form things called pseudopolyps. When eggs get swept into the liver, they cause hepatosplenomegaly, which is an enlargement of the liver and spleen, and a specific kind of scarring called periportal fibrosis. And then with S. hematobium, eggs in the bladder wall cause hematuria, which is blood in the urine, and chronic inflammation that can tragically lead to bladder carcinoma.
And there is another devastating and so often overlooked consequence of urogenital schistosomiasis. When the eggs get lodged in the reproductive tract, it causes a condition called female genital schistosomiasis, or FGS. This affects millions of women. It leads to pain, infertility, and it creates lesions that can actually increase the risk of HIV transmission 3 to 4-fold. It is a massive, hidden public health burden.
So, if this disease is so widespread and so damaging, you'd think it would be easy to find, right? Well, that's actually a huge challenge, especially in that chronic phase where the number of eggs a person is excreting can go up and down, making the infection really tough to spot.
So the classic method is just looking for the eggs in stool or urine. The Kato cot smear is kind of the workhorse out in the field. It's cheap, it's simple to do. The big problem, though, is it's not sensitive enough to reliably catch light infections. The formal ether concentration method is much better for that, but it needs a centrifuge and a proper lab, things that often just aren't available where the disease is most common.
But thankfully, diagnostics are evolving. Newer tools like the point-of-care circulating cathodic antigen test, or POCCA, are a total game changer. Instead of trying to find the eggs, this is a rapid urine test that actually detects antigens that are produced by the adult worms themselves. Studies using this POCCA test have shown a much higher prevalence of infection than we ever saw with microscopy. It's giving us a much truer picture of this disease's real footprint.
And once you can actually map out the problem, you can start to fight it. So let's look at the strategies being used to control schistosomiasis right now, and a new threat that's starting to complicate the battle.
Look, it would be great if there was one magic bullet for this, but there just isn't. You need a multi-pronged attack. The main weapon we have is the drug called praziquantel, which is really effective against the adult worms, but that is not enough. You also have to have snail control to get rid of that intermediate host. You need improved sanitation to stop eggs from getting into the water in the first place, and of course, education. And as for a vaccine, well, developing one for a complex macroparasite like this remains a huge scientific challenge.
In this quote right here, it just nails it. You can hand out pills all day long, but unless you actually break the cycle of transmission by dealing with the snails and sanitation, you are never, ever going to truly eliminate this disease. You have to have an integrated approach. And just when we think we understand the enemy, it changes. There is a new and frankly alarming threat emerging, and that's hybridization. What happens is different schistosoma species, including some that usually only infect cattle, are mating with the human-infecting species. This is creating hybrid parasites with totally new traits, things like different drug sensitivities or maybe different host preferences. In fact, these hybrids are thought to be responsible for recent and really surprising outbreaks in Europe, specifically on the island of Corsica. And that leaves us with a final and pretty sobering thought. For centuries, schistosomiasis has been seen as a disease of poverty, mostly confined to the tropics. But in our world of changing climates and global travel, and with parasites that can hybridize and adapt, is this so-called neglected disease about to demand our attention on a much bigger stage?